My name is Joern Diedrichsen, and in the next 20 minutes or so, I would like to share with you some of the things that we've learned about functional imaging of the human cerebellum. And I want to show you some of the tools that we've developed to deal with the analysis of data coming from this interesting structure. Now, the human cerebellum is really quite challenging to image. And the main reason for that is that compared to the rest of the brain, it is a relatively small structure. It only occupies about 1/7 of the total brain volume. Okay. At the same time, it's important in a wide variety of functions, not only motor functions, but also cognitive functions like language, working memory, executive functions, and social cognition. Now, what makes it especially tricky is that, as you can see in this 190 micron scan that Marty Serino and I took from a postmortem cerebellum, the cortex, the cerebellar cortex, is extremely folded. So you kind of see here how tightly packed these individual folia are in the cerebellum volume. So this makes it very hard to resolve this surface. Now, Marty took the scan and he actually reconstructed the cerebellar surface. So this is the first complete reconstruction of a human cerebellar surface, and you can kind of blow it up. And then we can stretch it out to a flat map. And the flat map you get is about 15 centimeters wide, and it's about 1.20 meters long. And the reason for that is that the cerebellum is folded only in one direction, like a big accordion. So when you unfold it, you get this long, thin stretch. And it has about 80% of the surface of the entire neocortex. So it's a lot of cortical real estate where things are happening. But it is very tightly packed. Practically, that means that when we image the cerebellum, functionally, here, for example, shown for 1.5 millimeter resolution, we will not be able to resolve these individual folia. So that makes it problematic to use the surface reconstruction on an individual basis to now visualize data. Okay. Peter Bazin, who is also giving a talk in this workshop, will show you some of the attempts to kind of move closer to a real surface reconstruction. But for the purpose of my talk, I want to kind of focus on the tools that we've developed for volume-based analysis of the cerebellum, which I think for functional imaging probably is right now a kind of a good strategy. Now, so the first part of the talk is about this pipeline. It's called, it's publicly available and downloadable as the so-called suite toolbox, and has a couple of components about which I'm going to talk a little bit. And then in the second part of the talk, I want to talk about functional atlases. So the atlases that show you where you are functionally in the cerebellum. And I want to convince you that the current state of the art, namely to use the cerebellar lobuli, is not a very good way of delineating functional boundaries. That there are actually functional regions in the cerebellum that we can find, real functional subdivisions. And I'm going to show you some of these parcellations that actually show those. And in the end, I want to show you kind of a cool online resource that we have developed that you visualize these different functional atlases for the cerebellum. Okay, so the toolbox that we've developed, or the imaging pipeline, really starts with the first step, namely to isolate the cerebellum from the rest of the cortex. So it's an automated algorithm that we have rewritten a couple of times now and further developed. And that algorithm automatically takes a T1 base scan of standard resolution and separates the cerebellar gray matter and white matter from the cortical white matter and gray matter. Okay? So in the end, you have these cerebellar gray matter and white matter maps and an isolation mask. Now, what that allows us to do is to cut out the cerebellum from the rest of the brain and then normalize them into a standard template. The thing about the cerebellum is that it kind of always has the same structure. So it always kind of has the same branches on this tree here, but how it's located in the brain is actually quite different. So here, for example, this subject has a really kind of happy, smiling cerebellum with a kind of perky up cruise 1. And this cerebellum is a little bit more droopy and kind of sad. Now, what we can do is we can use a nonlinear volume-based deformation of the cerebellum to now bring all of these cerebelli into a standard template. Okay, and we're using here the Dartel algorithm in SPM. Now, the template that we developed is a high-resolution template that is created by a nonlinear morph, and we're calling it a spatially unbiased infratentorial template, hence SUIT. It's unbiased in that it's unbiased relative to M&I space. So on average, the location in SUIT space is going to be the same coordinate as in an M&I space. However, it's a slightly more high resolution template of the cerebellum. It has a little bit more spatial details, so it gives you a little bit better normalization than even the nonlinear whole brain templates that are around. Now, we also, for the SUIT normalized data and M&I normalized data, we have developed a surface visualization tool for the cerebellar data. It's kind of not a real surface. It's not the 1 meter 20 by 15 centimeter surface that I showed you in the beginning. It's kind of a approximate hull surface around the cerebellum. And we can take this surface and then stretch it out and flatten it out into a 2D representation where we made sure that the size proportions of the different cerebellar regions are preserved in this projection. And this is a really kind of nice tool to visualize cerebellar functional data, and I will show my data in this representation. And again, this is downloadable as part of the toolbox that I described. Now, when we developed this pipeline in 2006, the standard normalization in the field was kind of the SPM2 normalization. And you can see that back then, the SUIT normalization really advanced the normalization process a lot over that back then state of the art. Okay, so here you can see that if you take the primary fissure or the interbiventa fissure from 20 individual subjects and you project that into the atlas space, you can see that SPM2 did not normalize it very well. However, SUIT really managed to superimpose these fissures exactly in atlas space. In the meanwhile, many better normalization algorithms, volume-based whole brain normalization algorithms have come along. So you can kind of see SPM segment or FSL FNERT, and they really come much closer to the performance of the cerebellum. However, SUIT still has a slight performance advantage, and we think this is due to the isolation step. So SUIT really can concentrate on bringing the cerebelli into alignment rather than having to worry about the immediately abutting visual and inferior temporal cortex. Now, there's a second reason why the isolation step is so important for functional data analysis. And the reason for that is that if you record activity in the cerebellum, usually there's also some activity in occipital and inferior temporal cortices. And to show you the effect of that, what I've done here in this image is simulated very strong activity in inferior temporal and occipital cortex. And usually the T values in these regions are 2 to 3 times higher than the ones that you find in the cerebellum. Okay? So even though there's, in this example, simulated example, there's no activity at all in the cerebellum, if you take this activity and now, without any smoothing, project it onto the flat map, what you can see is this so-called cortical bleed, meaning cortical activity that is just leers down into these characteristic lobulus 5 and 6 areas. Okay? With smoothing, of course, this is much worse. Okay, so what our pipeline standardwise does for functional imaging data is therefore in native space already mask the functional data to only take functional voxels that really come from cerebellar gray matter, and then after masking, do smoothing and normalization and projecting onto the flat map. And this approach really effectively reduces this cortical bleed and avoids any artificial visual activity in the superior cerebellum. Oh, this is very important. Now, all these tools, the isolation, the normalization, the templates, surface-based visualization, VBM pipelines, and for example, other tools like tools for the normalization and atlases for the deep cerebellar nuclei are downloadable at this website. Now, this toolbox has been developed as an SPM toolbox. However, I should stress that it's not dependent on doing your analysis in the SPM pipeline. All these steps are pretty modular, and you can use it in your analysis pipeline of choice. We're also translating many of these tools now into Python to make it available to a broader public. Now, the Steele toolbox, and especially also this neonatal template that we have for the Steele toolbox, have been developed by Carlos Hernandez Castillo, a former postdoc in my lab, who's now starting his own lab at Dalhousie University. And he recorded a really great 12-minute video that gives you some introduction into how to use Steele and how to use these different tools. And you can watch it as part of this workshop. Okay, so now I'm coming to the second part of the talk, namely functional atlases of the cerebellum. Okay, so as I said, there are a lot of different functions crammed in the cerebellum into relatively small space. So it's very important to subdivide the cerebellum into different regions. Okay, now defining functional regions has a long history for the neocortex. So we have the Brodmann areas, we have many different parcellations. And so what we can do there is cut up the cortex into different distinct regions that have their own histology, their own connectivity, and probably their own function. Okay? Now, for the cerebellum, that's not as easy. And the main problem here is that the cerebellar cortex is very homogeneous across the whole cerebellar cortical sheet. There's always that standard 3-layer cortex. There are no real differences like you find in the neocortex. And so traditionally, what people have done is the only thing that they could do, namely annotate where you are in the cerebellum based on the lobulus. So the lobuli are these branches that came out of the white matter body here. So, and these are kind of preserved across subjects. Okay. Now, the standard lobular notation we really owe to Olaf Lassell, who was a comparative biologist who studied the cerebelli in birds and in various mammal species. And he found that across species, everybody had these 10 cerebellar lobuli. And Jeremy Schmaman and colleagues then published a very important MRI atlas of the human cerebellum. We took this notation and really applied it to the human. And this is kind of the standard notation that we have now in the field. So in this notation, lobulus 1 through 3 are very, very small in the human. 4, 5, and 6 make up the superior cerebellum. Lobulus 7 is the largest by far. It takes up 50% of the cerebellar gray matter, and it's subdivided in crus 1, crus 2, 7b. We have lobulus 8, lobulus 9, and then the so-called nodulus and flocculus in lobulus 10. Okay. Our group, to make this atlas available to the neuroimaging community, did the handwork of hand labeling 20 individual cerebelli using those notations, and then developing a probabilistic atlas that ensued an MNI space that you can download from the site for plugin for different neuroimaging packages like FSL, AFNI, or OHBM. Now, this atlas and this notation is widely used in the community now. So people describe where they found activity in the cerebellum, like I found activity in crus 1 or crus 2. Meta-analysis are being described as or summarized as these are the characteristics of task that activated crus 1 or crus 2. And people define for their studies regions of interest based on these lobuli. Okay. Now, the problem, and this is kind of one of my main take-home messages from this talk, is that it's not clear whether these lobular boundaries are actually useful boundaries for defining functional subdivisions. And I actually want to argue that they're not. Now, we wanted to look at this question. And we looked for a data set that really, where we could describe the function of different cerebellar regions across a vast range of tasks. And there were a couple of data sets there where a number of subjects were studied in a couple of tasks. There were a number of meta-analyses available across different tasks, but we didn't really have the comprehensive task data set that really would allow us to functionally map the human cerebellum. So what we decided was to pick 47 task conditions across basically cognitive neuroscience in general, so the whole kitchen sink, and study 24 subjects, scanning each subject about 6 hours each, and get a really definite data set on this. And this paper is published last year. Now, what the result of the study was that this is only a subset of these task data sets, that you see that we could elicit very clear and very, across the two experiments, very reliable activity patterns in each task in the human cerebellum, and that these activity patterns also really clearly differed across tasks. So you have clear, typical motor task, here's kind of social cognition task, and working memory, action observation. Each of these tasks really provided a different, very distinct activity pattern. Now, when you look at these activity patterns, it's not immediately clear that they are well delineated by the lobular boundaries, which are shown here by the thin white lines on the fragment. Okay. To evaluate that objectively, what we wanted was a good criterion to evaluate whether a functional boundary, however you want to draw it, actually separates functionally distinct regions. Now, an obvious way to approach this is just to compare how much two voxels within a lobulus or within a parcel correlate with each other. And they should correlate more than two voxels that lie between these two parcels. Okay? Now, when you do this anywhere in the brain, what you're going to find that the correlation, the functional correlation between two voxels will strongly depend on the distance, the spatial distance of these two voxels, with nearby voxels being pretty highly correlated, and the functional profiles of voxels that are further away, where the correlation drops off. Now, so this is just a function of possibly smooth functional gradients across the cerebellum and the cortex. Now, if you want to show that there's a real functional boundaries, you need to take this effect in mind and take it into account. So what we did here is to compare the correlation between, for voxels within a parcel and between parcels for a specific spatial distance. So we only took voxel pairs that were maybe 2 centimeters apart, and then compared the pairs that were between and the pairs that were within. Okay, and when you do this for the lobular parcellation, you can see that there's hardly any difference. Okay, the within parcel correlations are just slightly higher than the between parcel correlation, but it might just be due to the anatomical folding. Now, so the take home from this is lobular boundaries really are not very good functional boundaries. Now, the question is if there are functional boundaries in the cerebellum at all, right? Maybe it's just really better described by smooth functional gradients. So to test this, what we did was we took our task data set and used the functional profiles to cluster the cerebellum into 7, 10, or maybe 17 clusters. Here I'm showing you the 10 parcellations that we found. Now, when you look at this parcellation, you clearly see that these parcels do not fall along lobular boundaries. For example, when you look in lobulus 7, you see 5 medial to lateral regions here that span cruise 1, cruise 2, and 7b. So span basically lobular boundaries. Now, when you evaluate this parcellation on our task data set, you now find a substantial difference between the within and between parcel correlations for a given spatial distance. So this is now a much bigger difference here, indicating that these new parcels really are defining distinct functional regions in the cerebellum. Now, you might say, well, that's cheating because you derived your task-based parcellation on the same data that you tested on. So for that, we took a cross-validation approach where we defined the parcellation on one experiment and then tested only on the absolutely unique tasks in the other experiment. Okay, so the parcellation hadn't seen the task where it needed to predict the functional boundaries. This is the same thing as you would use a group parcellation to now apply to your own new task. And we can't expect that your task was actually in the training data set. Okay, and when we do this, you can see that the difference between the within and between correlation actually reduces just a little bit. Okay, so this is a very robust parcellation that is really predictive of functional boundaries across many, many different tasks. Now, the difference between the between and within parcellation is kind of a good metric of how strong a functional boundary is. And we're calling this, because we're controlling here for the spatial distance, the distance controlled boundary coefficient. And there's a poster here at OHBM by my graduate student, Chi, who is going to talk more about it if you're interested. Now we can take this difference, so the difference for the between and within, the distance controlled boundary coefficient, and plot it as a function of spatial distance for the lobular parcellation and for the task-based parcellation. And you can see that the task-based parcellation does much better in defining functional regions in the cerebellum. Now, there are other functional parcellations based on resting state data, most prominently the parcellation proposed by Joern and Buckner in 2011, where they labeled cerebellar voxels depending on the correlation with cortical resting state networks. Now, when you look at the Buckner 17 parcellations, you can see that there is actually quite a lot of common structure between our task-based parcellation and this resting state parcellation. Okay, there are other parcellations, and they actually also share some certain features. They all kind of look a little bit different, but there are some characteristics that are clearly shared. And when we evaluate the task-based parcellation, the resting state parcellations on our task-based data, we can again see that they are performing actually quite well. So overall, task and resting state parcellations are much more predictive of functional boundaries in the human cerebellum. So if you want to define functionally distinct ROIs for your analysis in the cerebellum, this is a much better approach than taking lobular ROIs. Now, as you can see from the graph, the task-based parcellation has still a small advantage over the resting state parcellation, and that's even though we cross-validated this evaluation both across subjects and across tasks. Now, another advantage of the task-based parcellation is that we can give each of these regions functional labels. So what we did was we took these 47 tasks and described them with 100 plus terms from the so-called cognitive atlas. So it's kind of a taxonomy that Russ Poldrack has created that describes different tasks by different cognitive processes. And so what I'm showing here for each of the regions are the three most important features that are common to the tasks that activate this particular region. You can download again this atlas from our website. Now, as the last thing in my presentation, I want to show you kind of a cool atlas widget that she, a graduate student in my lab, prepared that allows you on a website to browse both volume and surface-based representations of these functional maps. And I'm going to show you this now in live. So here's a viewer in a browser window, and you can see that there's this volume-based representation and a surface-based representation. The nice thing is that if you click in the volume, you can actually see where you are on the surface, and you click on the surface, you can see where you're in the volume. So you can kind of explore the flat map and the cerebellar anatomy in this viewer. You can now load different parcellations, for example, the Buckner 7 network parcellation here, and kind of explore where this is. You can explore also all the contrasts that we had in our functional imaging study. There is, for example, the action observation task. So you can kind of explore these 4 different distinct activation blobs that we found in the cerebellum for the action observation task. So this is a very useful tool if you find cerebellar activity somewhere and you really want to know what is this region, what characterizes this region functionally, and where is it located in these different atlases that you can that describe the cerebellum functionally. Now, I want to end my talk with thanking and acknowledging all the people who've done the hard work, especially Maeve King, who was kind of the lead author on the multi-domain task battery study. Carlos Hernandez said he has his own lab at Dalhousie University now, who is co-developing the Suite toolbox with me, and Dachi, who has prepared this online viewer and is also involved in many other activities around cerebellar analysis. And my external collaborators, Marty Serino, Richard Ivory, and Russ Poldrack. And I want to acknowledge my grant funding and thank you for your attention. All right, so one important question that I didn't get to address in my main talk is, what does bold signal in the cerebellum actually mean? And this is kind of a very important question when we're trying to interpret functional imaging studies of the human cerebellum. Now, luckily for us, we know quite a bit about where the bold signal in the cerebellar cortex comes from due to work from a Danish group around Martin Lauritzen, who over the last 50 15 or 20 years has really done a lot of elegant experiments looking at blood flow regulations in the mouse cerebellum. And what we learned from that is that vasodilation, so dilation of blood vessels, and ultimately leads to observable Bolt signal, really is triggered by a cascade of events that starts with mossy fiber input. And in the human, most of the mossy fibers actually rise from the neocortex onto granular cells, transmission to stellate cells, and then nitric oxide emission that leads to vasodilation. Now, at the same time, the Purkinje cells, so these are the only and major output cells of the human cerebellum, do not regulate blood flow seemingly at all. Okay, so what this group showed that you can double the simple spike firing rate of Purkinje cells by removing the inhibition from basket cells onto the Purkinje cells, and you basically see no discernible increase in blood flow. So other studies have shown that Purkinje cells do not elicit a cascade that leads to emission of nitric oxide that then leads to vasodilation. So taking these together, it's really a pretty good guess that when we look at Bolt signal of the cerebellum, we really pretty purely look at input, especially cortical input on the human cerebellum. So if you image the cerebellum and see activity, it's really mirrored cortical activity, how it arrives on the cerebellar circuit. Okay, so this is kind of an important constraint when we start interpreting cerebral imaging studies. So it's not the output; it's really very cleanly the input, and this is a much more dramatic and cleaner, probably cleaner dissociation than what we find in the neocortex.
Hi, my name is Carlos Hernandez, and today I'm going to show you the basic function of SUD toolbox and how you can use it for your cerebral research. SUD is a set of MATLAB functions that allows you to perform several operations in MRI images. Many people have sent emails asking for a recipe to use SUD. However, there is not only one way to use the toolbox, and that's what makes SUD a useful tool to analyze cerebellar data. SUD can work with different kinds of MRI data. However, we need to start with an anatomical image. This can be, for example, a T1 weighted sequence. In this graph, the orange boxes represent images and the blue boxes represent functions. We will use the isolation function to separate the cerebellum from the rest of the brain. This function will create a cerebellar mask as well as gray and white matter probability images. This operation is performed in the native space of your subject. Therefore, to perform any group analysis, we need to normalize our images to a standard space. Here we can use the normalization function, which creates a flow field that can be used to deform images from the native space to SUD space and vice versa. At this point, depending on your scientific question, you can use the reslice function in different kinds of images. For example, you can take a functional image of cerebellar ball activity and reslice it into SUD space to perform a group analysis. Another approach is to take a region of interest that was defined in SUD space and take it to the native space to localize such ROI in each subject. This can be used for functional or anatomical analysis. As you can see, multiple approaches can be followed. Finally, any image in SUD space can be transformed into a surface using the flat map function. This function will take the gray matter voxels in the volume and project them into a flat representation of the cerebellar cortex. Now let's check out how to use these functions in a practical way. Once you downloaded the latest version of SUD, be sure to place the folder in the right location, so SPM and MATLAB know where to find it. Although SUD comes with a graphical user interface, we will explain the functions using the command line. In this way, you can create your own scripts to automatize your pipeline. This is helpful, especially when you need to run several subjects. Before using SUD, we need to perform some preprocessing in your images. If you don't check these simple steps, you might run into problems. The first thing you need to do is to check that your images is in LPI orientation. If your image came out from the scanner in a different orientation, then they should be resliced. The second step is to set the origin of the image into the anterior commissure. This operation is necessary for the normalization. These two operations can be performed either in SPM or any other software. It is also important to remember that any image that you want to use for your analysis must be properly aligned to the anatomical image that you are using as your starting point. For this example, I will use a T1, a T2, and a functional image of one single subject. Before using Soot, I aligned all of them, therefore I can apply any deformation to any of the image with no problems. To perform the isolation, just type Soot _ isolate _ seg, open parentheses, and you need to specify the image or images that Soot will use. Open curly brackets and type the file name as a string. Don't forget the semicolon at the end and press enter. This will take about 3 minutes to finish, and you will have in your folder the cerebellar mask and the tissue probability images for gray and white matter. Remember that we need to check the resulting cerebellar mask, and if we find areas we want to remove, we should edit the image. One way to improve the isolation is to use a combination of MRI sequences that provide better contrast. For example, a T2, magnetization transfer, etc. To do this, you can just add the additional images when you call the function. Soot isolate seg, open parentheses, open curly brackets, type the first file name and then use the comma to separate the second file name. Here we will use a T1 and a T2 only. So we close the brackets and the parentheses, the semicolon and enter. All the functions of Soot Toolbox provide a description that can be visualized by typing help before the function name. As you can see, we describe the inputs and outputs, but also some optional parameters that the user can modify. The normalization is performed using the function Soot _ normalize _ dartel. This function requires a structure with the images resulting from the isolation. We can easily create this structure by typing the name and the fields. For example, let's say that we want to name our structure job. Then we write job.subnd.grade equals open curly brackets and we type the name of the gray matter segmentation. We close and then we do the same for the white matter. We save the name for the isolation mask and our structure is ready. Now we can call Soot normalize dartel parentheses job. Close and press enter. The function will take a couple of minutes and will produce two files, one containing the affine transformation and the second with the deformation field. As I mentioned before, from this point forward, you can use different functions depending on your scientific question. Let's imagine that you run an fMRI experiment and you have a first-level bolt activation maps for each subject, and you would like to perform a t-test between groups or even a simple group average. In this case, we can use the function Soot _ reslice _ dartel. We just need to create or modify our structure to include the proper fields. We can type job.sub.affinetr equals open curly brackets and the file name of the affine transformation. Then we will do the same for the flow field. And then we will include the images we want to reslice using the field resample. This function can receive several images in this field, so we can apply the same deformation to several functional images in the same line of code. We just need to separate the names using a comma. For this example, we will use only one. Finally, to erase any activity from regions out of the cerebellum, We can also define the cerebellar mask. We press enter and the function will generate a new image with the prefix W that now is in the SUD space. After applying this method to every subject, you can run any statistics in the normalized images. Another common application of SUD is to perform voxel-based morphometry analysis. In this particular case, instead of reslicing an activity map, we will reslice the gray matter probability map. The procedure is similar to what we did before. We are going to change the field resample in our job structure. We type job.sub.resample equals to the sec1 image. One important difference for voxel-based morphometry is that we need to specify that we want to use modulation in our images. In the previous case, the voxel values are preserved during the reslicing. However, for voxel-based morphometry, we want to modulate the gray matter probability based on the Jacobian. To use this option, we just need to define it in the job structure by typing job.jactransf equals 1. Once you apply this to all your subjects, you can run your statistics and create a voxel-based morphometry map. But what about moving things in the opposite direction? For example, we might have an ROI definition in pseudospace that we want to explore in a group of subjects. We can do this using the function pseud_reslice_dartel_inv. The procedure is similar to what we have seen before. We create a structure with fields for the affine transformation, flow field, the image we want to resample, and in this case, the image we're going to use as a reference. This usually is the original T1 image. The result is the ROI in the native space. This operation is an alternative that can save you a lot of processing time and space by keeping your functional images in the native space. Calculate the lobular volume still is a common practice, and I constantly get questions regarding this type of analysis. For this reason, I made this even easier for the user. If the function pseud_reslice_dartel_inverse is called with the field resample empty, the function will automatically return the pseud-lobular atlas in the native space of your subject. Then we can use the function pseud_vol to retrieve the volume of each lobule. Any image in pseudospace can be projected to the flat map by using the function pseud_map_to_surf. For example, we can create a variable map equals pseud_map_to_surf, we open parentheses and give the name of the image we want to convert. Then we can use pseud_plot_flat_map to plot the image. There are some options you can modify, such as the color map using the option CMap, or we can change the values in which the color scale is confined. For this, you can use the option C-Scale. Please review the flat map webpage for more examples. I hope that this video tutorial was helpful and that now you have a better idea of the different operations that you can perform with SUD. I also recommend you to read the documentation that we have in the website, where you can find more information about SUD and other resources. We thank you for the interest in using SUD, and please feel free to contact us with more questions and suggestions on how we can make this tool better for the community.
Hello everyone, and welcome to this educational session on the cerebellum. I am Pierre Bazin, and I will talk to you a little bit about cerebellar surface segmentation and cortical mapping. First, I would like to introduce a little bit one of the reasons the cerebellum is such a fascinating and important structure in the brain, looking especially at the evolutionary angle from this very nice review from Susanna Herculano-Hausel's work, where she could map the number of neurons that you could get in the cortex and the cerebellum, and see very nicely in the figure A on the right that they all line up fairly well in terms of proportions, and the human H on top is just having basically a lot more neuron than everybody else. But if you look then in B at the relationship between the brain mass, so the volume, and the number of neurons, you can see that the cerebellum is surprisingly small while surprisingly compact. And that's quite a very interesting feature, but it's also why we have had so much trouble working with the cerebellum in neuroimaging. So let's start maybe with a conventional high resolution already 3 Tesla image at 1 millimeter, looking at the cerebellar structures. You can see it's fairly blurry and it's pretty hard to visualize. If you can improve the resolution, and now you can with high-end techniques, for instance, the HCP sequences also at 3 Tesla, you can go to about 700 micron, 0.7 millimeter. You start seeing a lot more the folia, the lobule separations. You see the branches of white matter. It's quite nice. If you happen to have a 7 Tesla scanner, you can even push it a little further to about 0.5 millimeter isotropic, and then you get also a lot of very fine details. Unfortunately, if you really want to start looking at the details of the cortex, you really have to go to post-mortem imaging, and even in this case, the histology as with a big brain data set, here resampled at 200 microns, which then finally gives you enough resolution to start looking at the surface itself. So here you can see a reconstruction of the big brain cerebellum on the surface level. So you can see here really, so the question, can we get to the cerebellar cortical surface? With MRI in living people nowadays, no. But what we can do is we can create a surface that is enclosing the cortex and separating the different lobules from each other. And that surface can be then very useful for processing. So this is what I will be mostly talking about, giving you details about how to perform cortical segmentation and surface reconstructions from 3T and 7T MRI, and then giving you a few examples on how you can combine that with lobule segmentation, atlases, surface-based mapping of structure or function in order to get further with cerebellar neuroscience. I will embed all these examples in the NIRES toolbox, which we recently developed for neuroimaging at high resolution. So focusing primarily on 7 Tesla imaging, but also with different type of MRI and also postmortem histology. There's a poster on that if you're interested in more details. And so let's start with the segmentation. So assuming we have a very nice 7 Tesla image at 0.7 millimeter isotropic. First, we have to remove the skulls and other extracranial tissues. And then we need to first do a brain segmentation that allows us to separate the cerebrum from the cerebellum. So not just the tissue types, white matter, gray matter, CSF, but also having some information about the different regions. It's particularly important for finding the boundary between the cerebellum and the visual cortex, which are very close to each other. In the NIRES toolbox, we use the MGDM algorithm, which allows us to separate these and maintain topological constraints between them. Once we've done that, we can then use the Cruise algorithm, which I will detail a little bit more later, to obtain a cortical surface segmentation. And from that, we obtain surfaces from left to right white matter, gray matter boundary, showing you the branches of the white matter going into the lobules. In the middle, a central average surface that is very useful for further processing that I will describe later. And then on the right, the gray matter CSF boundary surface, which corresponds more to what we expect from the cerebellar anatomy, showing you the exterior architecture and the lobules in the cerebellum. Now, we don't necessarily all have nice 7 Tesla scanners available, and a lot of the good and very nice neuroscience that we do nowadays is still with 3 Tesla imaging. And what happens if we have 1 millimeter 3 Tesla images? Well, we can still process them with very similar pipelines. So here again, we do skull stripping. In this case, we need to also do inhomogeneity correction. And then we can do brain segmentation again with MGDM. One thing of note here is that because we're at 1 millimeter resolution, things are a little coarser. So we can use a very simple trick, which is to resample the data to 0.5 millimeter after the segmentation to just focus on the cerebellum, for instance. And another interesting trick here, because we had to do inhomogeneity correction to remove some of the B0 inhomogeneities, it's often a very good idea when you do that to do a second pass of correction on the structure of interest. So here, just focusing on the cerebellum that was defined in MGDM and doing a second step of inhomogeneity correction. And finally, the cortical segmentation with screws. This is the surfaces we obtain again. So the white matter boundary, the central surface, and the CSF boundaries. And you can see, doing a comparison of the 2 types of surfaces, we have some fairly similar results. So 3 Tesla 1 millimeter MRI is already pretty good to get you a fairly detailed surface of the cerebellum. Now, in terms of practical computations, let's look a little bit at a few lines of code. So the neur toolbox is a Python toolbox. So you need to install it. You can look at the documentation and read the docs to get some details on this. But once you have it, you import it. And then you need to define your data set. You can download some of the examples that we have provided online. And then you run the different steps. First, the skull stripping, then the MGDM algorithm that separates the brain into major regions, and then a step of brain extraction, which prepares the data set to obtain the Cruise algorithm. So Cruise will work on any kind of data that would correspond to a cortex, cerebral cortex or cerebellar cortex. So first from MGDM, we have to extract the white matter, gray matter, and CSF membership functions for the cerebellum, and then we can then pass this into Cruise. So Cruise is a very nice algorithm. Well, I'm biased maybe, but I find it very nice. It was developed by Joern et al. In 2004. And the original goal was to segment the cerebral cortex with it. It has 2 main components. The first one is given some tissue probability maps for the white matter, the gray matter, and the CSF to create an anatomical enhancement of the sulcal regions. One of the challenges of cortical surface mapping, surface segmentation, whether cerebral or cerebellar, is the fact that inside the sulci or in the fissures between lobules, there is a lot of partial voluming, and the CSF is very difficult to estimate. But because the structure of the cortex is regular, you can make a fairly simple assumption that whenever you have branches of white matter around some region, then in the middle of that region, you should have a skeleton of CSF. So this is what the anatomical enhancement step does. And it will then create a map that is highlighting as well all the sulci regions in the cortex or the fissures in the cerebellum. From that, we will use a level set representation of the surfaces, I'll detail a little bit later, and create a successive surface evolutions of that surface from white matter into white matter gray matter interface, and then gray matter CSF interface. The level set is a very convenient representation of surfaces that, instead of using meshes with complex mesh topology, will instead do an implicit representation based on the image domain. So in the image domain, you can define for each voxel the distance function to the contour of interest. If you take a sign distance function, making it negative inside the object and positive outside, for instance, you can then recreate the function, the surface exactly up to the resolution of the voxels. So what is nice with that is you can just go back and forth between the two. You're working in image domain, so it's very convenient to work with data that you want to map, for instance. And whenever you need a mesh, you can use the connectivity consistent matching cubes algorithm that will create a very high density, very high quality 3D mesh with subvoxel mesh sizes. Once we have those meshes, we can then do very classical things that were developed, for instance, for cortical mapping of the cerebellum, for instance, inflation. So the principle of cortical inflation is very simple. You do a mesh-based relaxation where you try to average the location of the vertex with the ones in the neighborhood, and then you just iterate over time until your mesh has obtained the required smoothness. You can see it from this algorithm from Tosun et al. So we can use this exact algorithm from cerebrum to the cerebellar cortex and obtain also inflated triangular meshes, which we can inflate up to obtaining some sort of a bean or keep a little bit of structure in it. By doing the intersection of the inflated mesh and the original mesh, then you can also obtain a nice representation of the fissures like you're used to see in cortical mapping. So with screws, you obtain those different meshes. So the white matter, gray matter interface, the gray matter CSF interface, a central surface, which is essentially the average of those two surfaces, and then the inflated version. You can see from these examples of 3D data that it works fairly well on a lot of different subjects, with a lot of the lobules being defined in those structures. If you compare it to 7 Tesla MRI, you can see that the overall architecture of the surface is very similar. The main difference between the two is that the 7 Tesla average surface is a little inside the 3 Tesla one, because at 7 Tesla you see a little better the CSF because of lower partial voluming happening. And so the CSF gray matter interface tends to be a little further inside the structure of interest compared to 3 Tesla. Again, let's look a little bit at how to code this. So on top, the first line, you have the cruise algorithms that we just ran. And now you need to build meshes. So you just create the meshes based on the level set for the gray matter, white matter boundary, CSF gray matter boundary, central surface, which are all outputs automatically from cruise. And then you can use surface inflation to create your inflated mesh with the desired level of smoothness. Now, if you've been going to the OHBM brain hack, which I highly recommend, you might want also to see what you can do around this example to fit different images or different questions. You may want to do your segmentation, your pre-segmentation with FreeSurfer, or use Suit to define your cerebellum, white matter, gray matter, and CSF. So that is not a problem. Cruise will take those maps systematically. It just needs a topologically spherical regions inside the white matter to start from to create spherically, topologically spherical structures, and then probability maps to guide the automated surface estimations. Likewise, the cortical surface inflation, I use it on the central surface because it's the one that has the least amount of curvature among the three, but you could use it in any of them and get similar results. Additionally, I would like to point out that neur has a few interesting tools for surface processing, bringing mesh to level sets, so going into the other direction, creating level sets from probabilities, mapping, we'll see that a little bit more in detail. We have some filters that are very useful to better define the regions where you have a lot of partial voluming, so the fissures or the white matter branches. Topology correction, if you want to create a structure that has spherical topology, if you don't have it to start with. And also some laminar fMRI tools like volumetric layering, laminar smoothing, or profile meshing that allow to do some more advanced processing, looking at different depth in the surface definition. So in terms of applications, the first thing we would want to do is to start putting labels on cerebella. And if you followed the talk from Joern Diedrichsen, you've seen, for instance, the beautiful toolbox of Suit that does a lot of that very well. So when you have a surface, the nice thing is you can do that a little bit better because the volumetric labeling is very challenging at the fissures. But the central surface we're creating then is further away from that region. And so when you intersect labels from, for instance, Suit, or in this case here, a co-registration of the chroma atlas that we created with the surfaces, you get a better definition of the lobules because they're defined based on the distance of the lobules on the surface rather than on the volume space. In addition, the surfaces can be very useful for co-registration with a lot of precision. We have a poster that you might be interested in that looks at how to do the surface registration with the surface approach in the cerebral cortex. But the same method can be used in the cerebellum. The cerebellum actually has a little less variability in general, and so the problem there is a little simpler. But the basic idea is to start from the central surface level sets, and with a highly optimized version of the Hans algorithm based on that information, we can create group averages of multiple subjects that actually look like single subject brains. Last but not least, if you have some high resolution quantitative MRI, for instance, with 7T, like Wietske van der Zwaag nicely presented in her talk, you can start looking at how those are distributed on the cerebellar surface. In the volume, things being very packed, it's very hard to find patterns. Whereas if you look in the surface, like we did here, you can start seeing a few different things happening, looking from inner to middle to outer cortical boundaries, and looking at T1 and T2 star patterns, respectively, here, left and right. You can start seeing a little bit some separations that looks like bands on the T1, and also quite strongly in the T2 star, you can see very strong separation of the upper and lower regions. So in order to do that, with the code, it's again a few lines of code. So if you have to co-register data, you need to use a ANTS co-registration. We have some embedded version that is nicely integrated with the neur system. Then you can just transform the lobules or any other information you wish to map onto the cortex. And then you just do a surface mesh mapping that will intersect the volumetric data with your mesh. If you have data that is already co-registered intrinsically, like the quantitative T1, then you just need to do the surface mapping. So in summary, I hope I could show that cortical surface reconstruction for the cerebellum is feasible. It's feasible at 7 Tesla, but also at 3 Tesla. However, you have to be very clear that it is not going to follow the cortical folia. There you need at least 200 microns, maybe a bit more. But that provides a lot of enhancement for separating neighboring lobules. It also allows you to do cortical inflation, mapping, like in the cerebrum, and to have a different way of visualizing information that is very difficult to see in the cerebellum because everything is so tightly folded. You can do single subject and group analysis using the surface for basis for registration. You can play with laminar tools, and I would argue that the neur toolbox is quite well adapted to this type of things because you have Python modules which you can interact with a lot of different software packages. Everything can be done in the image space thanks to the level set representation. And you also have a lot of additional tools for 7T imaging and high resolution neuroimaging. With that, here's a list of references, and I would like to thank you for your attention.
Hi there. So hopefully at this point you've heard from other people in this amazing educational session about how to acquire and analyze and localize some of your cerebellar neuroimaging results. And what I'm going to talk to you about today is the final step in the research process, particularly how we interpret these cerebellar results and how we apply them to the study of language and neurodevelopmental disorders. And I'll focus on these things because the role of the cerebellum in motor performance is relatively more established, and also because these are areas of research that are interesting to me. And so some of the main questions that I'll be talking about today are, first, how do we interpret cerebellar results once they've been acquired? And I'll share with you some of the examples of our own research that really spurred us to look at location and specificity, and specifically drill down to the level of functional subregions when it comes to cerebellar analyses. The second thing I'll talk about is how can we examine complex linguistic behaviors within the cerebellum? And I'll share with you again some of examples of our work that have tried to move beyond location and subregions within the cerebellum, using behavior, interactions between cerebellar subregions, and also cerebrocerebellar circuits. And lastly, I'll talk a little bit about what studying the cerebellum can add to our understandings of neurodevelopment and neurodevelopmental disorders. And we'll talk about how in this research domain in particular, you should ignore the cerebellum at your own risk. Okay, and so just broadly, the cerebellum is anatomically divided up into a series of lobules, and they're numbered here. And for a long time, we've known that the anterior lobe and regions down here are associated with motor tasks and motor performance. But the past few decades of research have shown that these large posterolateral hemispheres in the cerebellum are actually more important for cognitive processing. And in fact, these regions, the majority of the cerebellum, in fact, is functionally and anatomically connected to association cortices, non-motor regions of the cerebral cortex, including regions that are important for things like abstract thought, language, and cognition. And over the past few decades, we've learned that the cerebellum definitely does contribute to language, but that strikingly, the cerebellar contributions to language are not only vast, but quite varied. And they range a lot. So at one end of the spectrum, the cerebellum houses the motor effectors for speech. These are things like the tongue and the mouth. And at the other end of the spectrum, the cerebellum is also involved in things like semantic processing or verbal working memory. And as you can see from some of the examples I've picked out here, different regions, discrete regions within the cerebellum, have been associated with a variety of tasks over the past few decades. And until quite lately, what a lot of researchers did was try to localize specific tasks to specific regions of the cerebellum. This was often done taking a lobular approach, now our understanding of how to localize results has actually started to change. And this actually occurred quite early. As you can see, activation for things like language or things like motor behavior in the cerebellum aren't restricted by these lobular boundaries that I mentioned before. In fact, they span lobular boundaries and occupy large swaths of the cerebellum. And over time, our understanding of this cerebellar functional topography has really evolved, so much so that now we can actually reliably map discrete sub-aspects of language, things like verbal fluency or word comprehension, to very specific regions within the cerebellum. And what's also important to note is that this organization of subregions or networks within the cerebellum may actually reflect anatomical realities. So for example, molecular patterning or zebrin patterning, for example. And these are things that are intrinsic to the cerebellum and may not solely rely on how the cerebellum is connected up with the rest of the brain. And so hopefully at this point, I've convinced you that, localization within the cerebellum is important. And I want to share with you a few examples of how we've tried to drill down into individual functional subregions within the cerebellum to really elucidate how it contributes to language and cognitive behaviors. So the first is an example that we recently published, and we were interested in examining cerebellar contributions to reading fluency. And so for this task, participants came into the fMRI scanner and they saw words that were presented sequentially, and these words formed a sentence that was either semantically plausible or semantically implausible. And what was interesting was that we manipulated the rate at which these words appeared. Sometimes these sentences were presented quite fast, and other times they were presented pretty slowly. And we were interested in the interaction between semantic plausibility and presentation rate. And what we found was that these regions of the cerebellum were engaged primarily when sentences made sense, but when they were presented at a fast rate. And so we think these are regions of the cerebellum that are important for maintaining comprehension in the face of taxing fluency demands. But what I'd like to point out is that these clusters both of them occur at the border of similar networks. So they all occur within motor, attention, and executive control networks. Despite this, however, when we looked at behavior, only one of these clusters actually showed a correlation with standard measures of reading fluency outside of the scanner. And so this was interesting to us for a few reasons. One is that behavior really elucidated specific contributions of these subregions despite being localized in the same network. And the second was that, though of course location in the cerebellum drives function, there's specific ways in which location in these subregions may contribute to a specific linguistic task. And so moving forward, we've tried to look beyond just at the network level at individual subregions, and we've also tried to use behavior to elucidate the contributions of specific subregions within the cerebellum. One other thing we've noted is that just like in the cerebral cortex, the cerebellum has two hemispheres. And we've noted in our work that lateralization actually matters within the cerebellum, in addition to subregion specificity. And so here I'll show you an example of work that I contributed to as a graduate student in Catherine Studley's lab. And this was work that was done in collaboration with Pierre at UT Southwestern. And so Peter Tsai has an animal lab, and he took mice and placed them in a chamber with both a novel animal or a novel object. And this is a measure of social approach. Typically, mice will spend a lot more time exploring the novel animal than they will the novel object. And what we were interested in is chemogenetically targeting CRUS1 or regions within CRUS1 in the cerebellum to see what happened to these social approach behaviors. And what was interesting was that if we inhibited right CRUS1, or regions within right CRUS1 in the cerebellum, we could abolish this typical behavior, make it so that animals no longer distinguished between the novel animal and the novel object. On the other hand, if we did the same kind of inhibition in left CRUS1, there was no such effect. The animal continued to act typically. And the same was true with cerebellar rescue in a mouse model of autism. This mouse model of autism doesn't differentiate between novel animals and novel objects, as you can see here in the control condition. But when stimulating right CRUS1 of the cerebellum, we could rescue this behavior and make the animal pay more attention to the novel animal and its environment. And this same rescue was not possible when stimulating left CRUS1. Okay, so hopefully I've convinced you now that it's not enough to merely say that results occur within the cerebellum, and it's not even enough to say that results are occurring necessarily within certain networks within the cerebellum. We should be specific about the subregions within which we're seeing our results, and also take into account things like lateralization. But something I'm interested in is how we can examine complex behaviors within the cerebellum. So as I showed you before in this schematic, a lot of previous research has used really discrete tasks to assess cerebellar contributions to specific aspects of language. But complex language, for example, semantic processing, requires a lot. It requires not only abstract processing, but also concrete processing. And so what we've discovered is that there's a spectrum of abstract to concrete processing that may underlie successful performance in any of these more complex linguistic tasks. And so can we capture this spectrum of concrete to abstract that is necessary in complex linguistic tasks? And so I'm going to talk to you about one example of a way that we've tried to approach this. And this was a project that I did in collaboration with Derek Nee, who's a professor at Florida State University. And Derek had developed a task to look at hierarchical processing in cognitive control and this spectrum of concrete to abstract within the prefrontal cortex. And so we used his task, which I won't talk about too much today, to examine whether the same was true in the cerebellum. This task is a cognitive control task using linguistic stimuli. And just to give you some intuition for why this kind of thing is important, I'll give you the example of conversation. And so typically if you have a conversation between two individuals, there's actually a lot that goes on to support this complex behavior. At a very abstract level, individuals have their past experiences, which they're going to draw on in the current moment in any conversation. But individuals also have really specific goals that they have to keep in mind that they're trying to attain via this conversation. And in addition to this, at the very opposite end of the spectrum, we have to pay attention to really concrete features in our environment, things like the eyes, the lips, the sounds that people are making, in order to optimize our actions to respond in a conversational settings. So there's obviously very concrete things happening as well. And lastly, every conversation occurs in a specific context. This might be a specific era or a specific location, but that context is going to drive how we use our abstract goals and experiences to drive concrete behaviors and action in a given moment. And so we use one task to look at all three aspects of this kind of complex processing. And what we found was that really discrete aspects along this spectrum activated really different regions of the cerebellum. And this recapitulated a lot of what we've already known or already seen from previous researchers, even within this own session, that have used discrete tasks to localize more concrete versus more abstract aspects of language functioning. And so in particular, we found that more abstract aspects of cognitive control processing using linguistic stimuli in this example activated these posterolateral regions of the cerebellum, and that more concrete processing activated regions that abutted sensory motor regions of the cerebellum. And of course, we looked at how behavior was associated with activation in these regions. And what we found was that activation in regions that were interested for this sort of abstract processing was associated with how we behaved in the future, but not how we behaved in the current moment. And the opposite was true for activation in these concrete processing regions. Activation here was associated with proximal behavior, behavior in the current moment, but not with preparing us for any sort of behavior in the future. But what I find really interesting is that complex behaviors probably require complex interactions between all of these regions that are undertaking specific aspects of a task. And it's possible that just like has been examined in the PFC, these regions aren't acting alone in the cerebellum. And in particular, we hypothesized that abstract regions probably drive activation in lower level concrete regions, and that this interaction between the regions is mediated in some sense by context. And that's exactly what we found. So we found that these regions aren't just discretely processing aspects of a task, but they actually rely on each other, both in terms of activation, but also in terms of future and current behavior. And so I'm going to pause here for a moment just to bring up one thing that cerebellar cognitive researchers often face. And that is that if you're interested in doing cerebellar cognitive or linguistic research, and you're about to respond to peer review, a typical comment that I and many others have received is something along the lines of, okay, we see your activation within the cerebellum, but how do you know, given connections between the cerebellum and the rest of the brain, that what you're seeing isn't a mere reflection of processing that's actually occurring in the real cognitive area, the prefrontal cortex, the de facto cognitive area? And so as a cerebellar cognitive researcher, we've started to think about how it is that we may creatively show that the cerebellum, of course, complements cognition that's happening everywhere in the brain, but also contributes something specific. And so first, I'd just like to say that the cerebellum isn't merely a reflection of cortex. And I'm going to share with you an example of how we specifically have tried to elucidate that. And so one thing that we did was we used an unbiased method, principal component analysis, to extract principal components from the prefrontal cortex and parietal lobe. And we regressed these components out of the cerebellum, and we saw what happened to interactions between cerebellar subregions when we removed the influence of cerebral cortex. And what you can see in this yellow and magenta lines over here is that it takes quite a bit of principal components. You have to regress out a lot of the influence from the cerebral cortex before the interactions between subregions in the cerebellum start to fall apart. And what's interesting here is that we can actually do this exercise in reverse. You can also take principal components from the cerebellum and regress them out of the prefrontal cortex. And you can look at what happens to well-established subregion interactions in the prefrontal cortex. And what happens is quite interesting. It actually takes much fewer principal components regressed out before you start seeing that these interactions in the prefrontal cortex are rendered insignificant. And so in a practical sense, this shows us there are creative ways that we can start to try elucidating what the cerebellum might be doing specifically or intrinsically. In our case, we looked at subregion interactions. And in a scientific sense, it shows us that the cerebellum is actually contributing something specific to cognition, and we're interested in exploring that further. Okay, and so the last thing I want to talk to you about today is the cerebellum and neurodevelopment. And as it turns out, the cerebellum is really important for neurodevelopment. Some previous evidence has shown that cerebellar contributions are actually more important earlier in development than they are later in life. And so this makes the cerebellum a rich place to explore when we're thinking about children and development. And some of our most striking findings have actually occurred in this research domain. And so I'd like to tell you or implore you to ignore the cerebellum at your own risk in this research domain in particular. And so one example of this is a study that we conducted in collaboration with Rachel Romeo, who was at the time a postdoc at MIT. And what we found was that when recruiting a group of 4 to 6-year-old children, so these are young children from diverse socioeconomic backgrounds, children were asked to pay attention to stories that occurred in either one ear or the other. So this was clearly a task that required a lot of top-down attentional control. What we found was that specific regions of the cerebellum are engaged by this kind of task. But what's especially interesting in this case is that the cerebellum and activation on this task is vulnerable to environmental influences. In particular, the amount of adult words an individual child heard was related to how active the cerebellum was in these children during this task. And so just to give you some intuition for this, I can pull out 2 kids. One is a kid who heard about 6,000 words a day at their peak hour versus a child who heard only about 3,000 words. And you can clearly see that engagement of the cerebellum on this task differs depending on the linguistic environment that a child is exposed to. And so clearly, if the cerebellum is involved in typical language development, it may also be involved in development in neurodevelopmental disorders. And one of my main areas of research has been exploring contributions of the cerebellum to autism in particular. Autism is a disorder that is characterized by social and communication impairments, as well as the presence of repetitive motor behaviors or restricted interests. And what many have found, and this is a figure from Sam Wang and Alexa Badura and others, is that damage to many regions of the brain can result in an autism phenotype, but only damage to the cerebellum at birth results in upstream, long-term autistic phenotypes over a lifespan. And in fact, cerebellar injury at birth is one of the leading risk factors for obtaining a diagnosis of autism later in life. And of course, we know that the cerebellum itself is involved in both motor behaviors and social communicative behaviors. And so this makes the cerebellum a really interesting place to look at in autism. And we were interested in language impairments in particular. Language impairments are some of the first signs of autism, and they result in long-term challenges with other types of behaviors throughout the lifespan. And what we found in one study was that decreased cerebellar gray matter volume is associated with later age of language onset. And you can see the regions that are involved here and the correlation down below. And what was especially interesting about this study was that in our sample, this was the only region in the brain that showed this kind of association. And so had we ignored the cerebellum, as many do, we might not have seen such a striking relationship. And so, again, I'd like to implore you to, in your research, ignore the cerebellum at your own risk, particularly in domains such as language and domains such as neurodevelopmental disorders. So I'll end with just some brief conclusions or takeaways. Obviously, we know that the cerebellum is implicated in a wide variety of language tasks. It's a rich place to look if you're interested in cerebellar imaging. When it comes to application or interpretation of results, what we found is that, of course, location within the cerebellum reflects function, but cerebellar activation is not constrained to lobules, and it's not constrained to broad networks or broad parcels either. And we should really explore this to understand what subregions within the cerebellum are doing. And in fact, specificity within the cerebellum, whether it's lateralization or within specific regions, says something about behavior. Importantly, the cerebellum does contribute something apart from cortex, and there's important and creative ways that we can try and get at how the cerebellum is contributing something apart from cortex. And lastly, I'd like to remind you to ignore the cerebellum at your own risk. There are certain research domains for which the cerebellum may be especially important, such as neurodevelopment and disorders. And so I'll just end by acknowledging my collaborators and mentors and my funding sources, and wish you happy cerebellar neuroimaging.